session 2 had to do how the microenvironment and the interactions that take place between CLL cells and other cells in the microenvironment, how they affect the survival, growth, and trafficking of CLL cells in a person. There were 3 speakers. The first was Andrea Pepper from the United Kingdom. Andrea spoke about the importance of signaling through Tolllike 9 receptors, and how that affected the biology of the cells, and in particular the trafficking. And her data looked like they may have an important relevance to predicting in advance patients that might do well or not on Ibrutinib therapy, or maybe better or worse on Ibrutinib therapy. The second speaker was Dimitar Efremov from North Macedonia, who is stationed in Trieste, Italy. And he spoke about very elegant studies that he had done and his team had done, studying the development of Richter's syndrome in animals that are made transgenic for the expression of the TCL1 gene, and studied how the elimination of certain genes that control, that are negative feedback genes, control the growth of these cells. And essentially what he found was that signaling through the B cell antigen receptor, IgM, surface membrane immunoglobulin, was key in the evolution of Richter's syndrome. And that alone seemed to be sufficient, at least in his experimental model, to develop Richter's syndrome. So in this case, it appeared that the cells, when they transform to Richter's, because they lose the expression of these special genes, are becoming relatively independent of the microenvironment. The third speaker was Alan Ramsey, again from the UK, who had a series of experiments looking at the interaction between the CLL cells and the other cells in the microenvironment. One of the most provocative was the effects on CLL cells of a subset of cells that are not part of the hematopoietic system. And these are called fibroblast reticular cells. In the normal setting, these cells are responsible for serving as essentially pathways or tracks around which CLL cells or normal B cells or normal T cells can move. And what he found was that the fibroblast reticular cells are abnormal or are made abnormal in a patient who develops CLL. And that abnormality affects the ability of the immune system to respond to the CLL cells in a normal way.